Hello everybody, welcome back to another AP Biology video. In the previous video, we discussed the process of photosynthesis, how our plants and other autotrophic organisms are able to harness light energy and use it to fix carbon dioxide into sugars. And today, we're going to talk about what we can do to put those sugars to use in generating cellular energy. We're talking about cellular respiration. The video today correlates with the Biology in Focus Chapter 7 on the same topic, and because it's a little bit of a longer one, I'm not going to leave any extra time here. We're going to jump right in. You may recall from previous units that living cells require energy. We have all sorts of things going on within our cells that need energy in order for them to occur. So within our ecosystem, the main source of energy is the sun. And when plants are able to harness that light energy coming from the sun, they're able to fix carbon dioxide into the building blocks of sugars like glucose. And then when organisms take in that glucose, or the glucose is used within a plant cell, we can use that to create chemical energy. So both products of photosynthesis, your oxygen gas and your glucose, are used to fuel cellular respiration. And the eventual product that we're going to get from cellular respiration is chemical energy, most typically in the form of ATP. Now we've talked a little bit about ATP already, so hopefully you understand that ATP is created through a phosphorylation event where we take ADP and an inorganic phosphate and we combine them together using ATP synthase, and that creates this energy carrier, storing that energy between the bonds of our second and third phosphate groups. When we look at cellular respiration, we are going to really break it down a little bit further because there are a few different catabolic pathways that are involved when we're talking about cellular respiration. We're going to mention a few of these now and then come back to them at the very end. But two of the ways that we kind of differentiate is either through the process of fermentation, where we break down our sugars, and this does not require our cells to have access to oxygen gas. So if you are an organism that lives in an environment where you have very little free oxygen, fermentation is one of the ways that you're still able to break down sugars and make just small amounts of ATP to get the jobs done. And then when we think of cellular respiration, really the main camp that we're looking at is aerobic respiration, where we're consuming our organic molecules and oxygen gas, and we're creating this large sum of ATP, cellular energy. Now, I would like to mention that when I say organic molecules, I mean all of our organic molecules. Your cells can use everything from carbohydrates, fats, and proteins as fuel, but we typically look at this pathway as using sugar in the form of glucose. So we're going to trace the process of cellular respiration using glucose as our stand-in for organic molecules. But it is important to keep in mind that we use all sorts of organic molecules that we get in our food to create this cellular energy. When we look at aerobic respiration as a whole, we're looking at a transfer of electrons from one step to another. We're attaching a lot of things to high energy electron carriers. In photosynthesis, we had NADPH. In cellular respiration, we have NADH and FADH2. And these both are able to carry an electron from one process to the eventual process that we will use to create that hydrogen ion gradient via our electron transport chain. So every time that we look at one of these energy carriers that kind of is produced through these earlier processes, we want to keep in mind that each one of those is carrying an electron that can then go on to make ATP. They're kind of the middlemen in this situation. If you're struggling to remember these energy carriers, I always associate NADPH with photosynthesis because photosynthesis starts with a P. And then our two that don't have that additional phosphate, NADH and FADH2, they come into play in cellular respiration. Now, when we talk about these electron energy carriers, it's important to recognize that they are the products of redox reactions. We've talked previously about redox reactions. In fact, I used this same slide where we talk about oxidation is a process where a molecule loses an electron, and reduction is where we've gained electron, and usually that's okay. Usually we're pretty good that far away, that go around. It's when we add in the other way that we can look at this that things start to get a little sticky. So we can also refer to things as reducing agents. So reducing agents are being oxidized, and oxidizing agents are being reduced. It's the nomenclature of what the activity is doing. So we kind of look at that in an opposite way. Reducing agents are electron donors. Oxidizing agents are electron acceptors. And so when we move our electrons from a less electronegative atom towards a more electronegative atom, we release all that chemical energy that can be used to put into work within our cell. So that is, in essence, what our energy carriers are doing. They are carrying this electron, and they are moving it to different areas, and then we can use that motive force generated by the electron in the electron transport chain to get all of that nice work, to push through our hydrogen ions into the interior of our mitochondrial membranes, and we can generate all of this ATP using chemiosmosis and ATP synthase. Back to the idea of organic molecules once again, we want to consider that when we have a lot of hydrogen, when we are highly saturated, we are going to have the most bang for our buck and able to break down these materials in order to produce the energy necessary to power cellular respiration. So we have fuel. This can be any of our organic molecules. We're typically going to look at it as glucose. You see glucose in its cyclic structure there at the top. But we oxidize glucose, so we remove electrons from it. And then at the very end of the process, we dump those electrons off into our oxygen gas, which is therefore reduced. Hydrogen also gets transferred to the oxygen in this process, and so that's where we get the water coming out of cellular respiration, and all that energy can be used to power our ATP synthase and our chemiosmosis to produce cellular energy. Now, the workhorses in this case are our electron carriers. Here is kind of a more in-depth diagram going through exactly what is happening when we are reducing these electron energy carriers. They can pull free electrons and hydrogen ions from the environment, from those molecules, and they can store them within their structure. They will be reduced. They are oxidizing agents in this case, and then they're just carrying them from one place to another. Now, the reason that we use electron energy carriers is because if we just burned the materials that are going into cellular respiration straight out, we would not get as much energy as we would if we created more of a stepwise process. When we burn just a straight fuel, we have a one payout. We get one sum of energy. But if we turn this into a series of steps and we pass our electrons to increasingly electronegative carriers, we create a series of energy releasing steps. And so we're getting controlled amounts of energy at a time. And the more energy we end up being able to harness in the later process. We're losing less as heat in this case. When we look at the pathways by which we harvest energy in cellular respiration, we are going to break that down into essentially 3 steps. Now, this can be divided up into 4 steps if we look at pyruvate oxidation or the link reaction as its own separate step. But everything starts with glycolysis. Every organism that needs to break down materials to produce cellular energy, they are going to start with glycolysis. Now, looking at that word, glyco referring to sugar and lysis meaning to break down. So it is in this process that we break our glucose down and we get a product known as pyruvate. Pyruvate or pyruvic acid needs to be broken down once more before it can really enter into the main process, the citric acid cycle, also known as the Krebs cycle. And so through pyruvate oxidation, we take this pyruvate and we modify it slightly. We attach it to a coenzyme and it's able to go into the Krebs cycle where we kind of turn the wheel a few times. We create a little bit of ATP, but really we're creating more of those electron carriers. And then all of these electron carriers can end up in oxidative phosphorylation. Oxidative phosphorylation, where we're taking those electrons that have been generated, we're using them to create that hydrogen ion gradient, and we're creating a large amount of ATP at the very end. Now, it's that final process, oxidative phosphorylation, that accounts for almost 90% of the ATP that we're generating from cellular respiration. We produce a little bit here and there in glycolysis and the Krebs cycle, which are what we call substrate level phosphorylation, because they are dealing with breaking down a substrate via a series of enzyme-mediated reactions. But it's when we get to the electron transport chain that we're really able to make a lot of that ATP molecule, that ATP energy. So we're going to get into each of the processes. I'll try to keep it a little bit more general for most of these because I do not need you to memorize the steps. I really just need you to recognize what goes in and what comes out of each of these processes. Okay, so we'll start our deep dive with glycolysis, where all of this begins regardless of if we are undergoing anaerobic or aerobic respiration. Glycolysis, termed sugar splitting, if we were to break down that word into its constituent parts, involves taking glucose and breaking it down through a series of reactions into 2 molecules of pyruvate, otherwise known as pyruvic acid. Now, along the way, we produce some high energy electron carriers as well as some water molecules as a byproduct. So a little bit of the water that we're producing from this overall process does arise via glycolysis. Now, the thing to remember is that glycolysis is an anaerobic process, so it's going to take place whether we have oxygen gas or not. If oxygen is present, then we can take all of the products that we've produced in this process and we can send them over to pyruvic oxidation, the citric acid cycle, and eventually oxidative phosphorylation. If oxygen isn't present, then we really can't do much with these products because we have no further steps in the cycle that we can really engage in. And so they'll have to take an alternate pathway, which we'll hit back to at the end of this presentation. Now, when we look at the process of glycolysis, it's important to recognize that there are essentially two phases. The initial phase, the energy investment, is where we actually have to put some cellular energy. Now, this is a drop in the bucket compared to the amount that we're eventually going to make. So usually cells are okay with giving up an initial little bit of energy in order to get a much greater amount at the very end. So this energy investment phase is followed by our energy payoff. So through a series of further reactions, we're actually producing twice as much ATP as we initially put in. So net, we're getting 2 ATP out of this process. We're putting 2 in, we're getting 4 out. So overall, we're getting 2 ATP from this process. And then in addition to that ATP, we've got a couple of high energy electron carriers that we produce that can go on to oxidative phosphorylation, and we can use those later on to generate more ATP. Glycolysis is a process that does not occur in the mitochondria. This should infer to you guys that glycolysis is a process that does not require mitochondria to be present, and so it does take place in a lot of single-celled organisms that don't themselves have a mitochondria, and it's likely ancestral. So we engaged in this process, I mean our cells did, prior to the acquisition of our mitochondria. Now I'm going to go through each of the individual steps through glycolysis, and we are going to see exactly how our glucose molecule goes from one molecule to 2 pyruvate at the very end. Our first step is that our glucose is phosphorylated by an enzyme called hexokinase, and this makes it more reactive and gives it a charge that traps the sugar within the cell, so it can no longer pass through the cell membrane. Good for us. This is our initial energy investment in this case. Following this, glucose 6-phosphate, which was our product in the previous step, is converted to fructose 6-phosphate by phosphoglucoisomerase. So if you remember that an isomer is a different form of a molecule, we have the same number and ratio of molecules. So we're kind of just doing some rearranging at this stage. This is followed by phosphofructokinase, which transfers a phosphate group. So we have a little bit of phosphorylation going here. This is a key step for the regulation of our glycolysis. So we have another ATP molecule that is entering into this process. It's dropping off a phosphate group, and we get a new product. Now, this molecule that we produce is cleaved by aldolase, and that creates 2 different 3-carbon sugars, and they differ in their orientation, so they are stereoisomers. We have DHAP and G3P. You may recognize G3P from photosynthesis and our Calvin cycle. Now, DHAP it undergoes one really quick reaction to convert it to G3P. So we're starting the rest of this process with the same material. And that happens almost immediately. So it's not like we have a buildup of DHAP anywhere within our cell. At this point, both of the G3P molecules are oxidized. An electron is removed and it is given to a high energy electron carrier. So we reduce our NAD plus and we form NADH, and you see that this is occurring twice. So we get 2 NADH out of this process. And the outcome of this is then further reduced, and we transfer one of the phosphate groups by an enzyme called phosphoglycerokinase, and that takes us to a more linear structure. And finally, well, almost finally, sorry, we got a couple more steps. Step 8, our phosphoglycer mutase relocates the remaining phosphate group. So we move it from the terminal to the central carbon. And through a little bit of water extraction, we produce a stereoisomer of that initial product. So this is called PEP. PEP, phosphophenol pyruvate. This is our essential first step of creating pyruvic acid. But we do need to get rid of that phosphate group. So we transfer that phosphate group one last time by an enzyme called pyruvate kinase, and we produce pyruvate. Now, my animation here is a little chaotic, so I have a little video for us on the next slide to kind of break down the process in a little bit of a simpler fashion.
Glycolysis, which begins the breakdown of glucose, is a series of 10 enzyme-catalyzed chemical reactions that can be divided into 2 main phases. In the energy investment phase, some ATP energy is used to start the process of glucose oxidation. By the end of this phase, a 6-carbon molecule, glucose, has been split into 2 3-carbon molecules of glyceraldehyde phosphate. The 3-carbon glyceraldehyde phosphate molecules now enter the energy payoff phase. Chemical bonds are broken, and NAD+ picks up electrons and hydrogen ions, forming NADH. The energy released is used to attach phosphate groups. The phosphates are transferred to ADP, finally making some ATP. This way of making ATP is called substrate-level phosphorylation. A couple more reactions rearrange the atoms in the 3-carbon molecules. More ATP is generated in the final reaction that yields pyruvate. For each glucose molecule broken down during glycolysis, a net of 2 ATPs are formed, along with 2 NADH molecules.
As mentioned previously, what we do with the pyruvate after we produce it from glycolysis does depend on whether or not oxygen gas is present within the cellular system. So we're going to follow in the case of oxygen is present. So in that case, our pyruvate, once produced, now enters the mitochondria, and we have a next step to really complete this process and get this molecule ready to go for the Krebs cycle. So pyruvate undergoes what we call pyruvate oxidation. So we remove a carbon molecule from our pyruvate. So this produces carbon dioxide, and we in that process remove one of the electrons. So we reduce NAD plus once more, produce another high energy electron carrier, and we attach the pyruvic acid byproduct to a coenzyme called coenzyme A, or CoA. So at the end of this process, we end up with an acetyl CoA, which can then start up our Krebs cycle. Now, the Krebs cycle, also termed the citric acid cycle, is what we're going to use to break down this pyruvate into the carbon dioxide that we release through cellular respiration. So every turn of our Krebs cycle is using one pyruvate. Remember, we have two pyruvates that we produce for every glucose molecule, so we actually get two turns in this whole process. So overall, everything that we produce in this one cycle, we can multiply by 2. So we're going to end up creating 2 ATP molecules, 6 NADH, and 2 FADH2. Now, FADH2 hasn't really been mentioned as much as NADH. It is another high-energy electron carrier, but it carries fewer electrons than we see with NADH, so it doesn't get used quite as much, more of an ancestral form in this case. So just like with glycolysis, we're going to walk through the different steps involved in the Krebs cycle. And remember, we do not have to memorize our steps. We just need to know what goes in and what goes out. So we will start with step 1. So the acetyl CoA that we've produced from oxidative phosphorylation, it adds its 2-carbon group to a molecule called oxaloacetate, and this produces a molecule known as citrate. So we've removed the coenzyme A, and we've attached those carbons to our citrate molecule. And this produces citrate. In the second stage, citrate is going to be converted to an isomer, isocitrate, and this is catalyzed by a contase. So after this step, isocitrate undergoes dehydrogenation and decarboxylation to form alpha-ketoglutarate, and this releases carbon dioxide and reduces one of our NAD plus molecules to NADH. Alpha-ketoglutarate undergoes oxidative decarboxylation to form succinyl coenzyme A, and this releases carbon dioxide and reduces another molecule of NAD plus to NADH. Now that succinyl CoA forms succinate in a reaction that's coupled with phosphorylation. So we get the phosphorylation of GDP to GTP. GDP and GTP are just like ADP and ATP, but we have swapped out the nitrogenous base in their structures. So we can almost read these as ATP in this case. They will eventually be ATP. From this point, the succinate is oxidized to form fumarate, and this reduces our other electron energy carrier FAD to FADH2. And then that fumarate can be converted into malate through the addition of water. And this is then dehydrogenated to form our original oxaloacetate, and thus reducing another molecule of NAD to NADH. Now that oxaloacetate, if you recall, was the first thing that we started with, so that can re-enter the cycle and we can start the whole process over again. You can see here that we end up producing 3 NADH for each turn in this cycle. We get 1 FADH2, and we produce 1 molecule of cellular energy in the form of GTP. Remember that we have to do this twice, so we end up doing 6 NADH and 2 FADH2 and 2 of our cellular energy molecules. Once more, I have a little animation here coming from the textbook that will help to kind of break down some of this process in a little bit simpler terms.
The oxidation of glucose continues in the citric acid cycle. Pyruvate molecules formed during glycolysis are transported from the cytosol into the mitochondrion, but pyruvate itself does not enter the citric acid cycle. A reaction occurs that removes a carbon atom, releasing it in carbon dioxide. Electrons are transferred to an NADH molecule, storing energy. Coenzyme A, or CoA, joins with the 2-carbon fragment, forming acetyl-CoA. 1 molecule of acetyl-CoA enters the citric acid cycle. The 2-carbon fragment of acetyl-CoA attaches to the 4-carbon molecule oxaloacetate in the first reaction of the cycle. This forms citrate. In a series of steps, bonds break and reform. 2 carbon atoms are released, one at a time, in molecules of carbon dioxide. Electrons are carried off by molecules of NADH and FADH2. One step produces an ATP molecule by substrate-level phosphorylation. A 4-carbon oxaloacetate molecule is regenerated. Since 2 acetyl-CoA molecules are produced for each glucose molecule broken down, a second acetyl-CoA enters the citric acid cycle. The same series of reactions occurs, releasing carbon dioxide and producing more NADH, FADH2, and ATP. The cell has gained 2 ATPs that can be used directly. However, most of the energy originally contained in the bonds of glucose is now carried by the NADH and FADH2 molecules.
So all of the steps that we've considered up until this point have been what we consider to be substrate level phosphorylation. And we've only really produced a net of 4 ATP molecules. Most of the energy that we've extracted up until this point is being carried on our high energy electron carriers. It's in this final step, the electron transport chain or oxidative phosphorylation, that we can donate all of those stored electrons and power ATP synthase. So we are going to look at the processes involved in oxidative phosphorylation here, not so much in depth because our electron transport chain operates very similarly to what we've seen already in certain parts of photophosphorylation of photosynthesis, but there are some differences. First, that our electron transport chain does require proteins that are embedded within the membrane. So we see that this is located within the inner membrane of the mitochondrion in our eukaryotic cells. For prokaryotes that engage in this same process, this is occurring in their plasma membrane. And so most of the components of this electron transport chain are going to be complexes of proteins with attached hemogroups that function as electron carriers. So we're essentially going to drop our electrons off and they will flow through the different proteins and use that motive force to create a hydrogen ion gradient. So the energy that is being released by this electron transfer, so with these high energy electron carriers dumping off their electrons, and then those electrons are flowing through the electron transport chain, we're using the kinetic energy of those electrons to move or pump hydrogen ions through into the inner mitochondrial membrane. And so this is going to allow our hydrogen ions moving from an area of high concentration to an area of low concentration to flow down its concentration gradient and power the turbine of that ATP synthase. The hydrogen ions can only cross the membrane through this ATP synthase. So we have this exergonic flow that drives the phosphorylation, and we get the process of chemiosmosis that we can use our hydrogen ion gradient to drive this phosphorylation process. So we're producing a whole lot of ATP as we push those hydrogen ions down their concentration gradients. The energy that's stored in the hydrogen ion gradient couples some redox reactions to this ATP synthase. And this is the concentration gradient that we have here is referred to as what we call a proton motive force. So this does emphasize its capacity to do work. Now, I did not go through and make a whole animation for this process, so what I do have for you guys is one last animation coming from the textbook to kind of break this process down.
Most of the energy harvested from organic molecules during glycolysis and the citric acid cycle is stored in NADH and FADH2. These molecules give up their high-energy electrons in the third phase of cellular respiration, oxidative phosphorylation, where most of the cell's ATP fuel is produced.
The electron transport chain is an array of molecules, mostly proteins, built into the inner membrane of the mitochondria. NADH gives up its high-energy electrons to the first complex in the electron transport chain. The electrons move from one member of the chain to the next, giving up their energy as they are pulled from NADH toward highly electronegative oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Oxygen captures the electrons in the very last step in electron transport. The last complex adds a pair of electrons to an oxygen atom and two hydrogen ions, forming water. The electron transport chain has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, stores the potential energy that was originally in the bonds of glucose molecules. The backed up hydrogen ions give up their energy when they diffuse through a special protein in the membrane called ATP synthase. As hydrogen ions flow down their concentration gradient, ATP synthase captures their energy to make ATP. This mode of ATP production is called oxidative phosphorylation because it is powered by the transfer of electrons to oxygen.
Most of the
Mentioned at the beginning of this journey, we were following the products as they moved through aerobic cellular respiration. So when oxygen was present, we could do everything that followed after the process of glycolysis. Now, there is a caveat that the majority of ATP that an organism creates is created through that final process of oxidative phosphorylation. But without oxygen to accept the final electron in that electron transport chain, oxidative phosphorylation will not progress, and we will not be able to produce ATP. So, when organisms do not have oxygen gas, they have to find other ways to generate ATP, since they can't rely on the electron transport chain and oxidative phosphorylation. So in both of the processes that we're going to look at, we are going to consider that glycolysis is still taking place. We're still breaking down glucose, and we're still producing 2 ATP nets from that process, and we're still getting pyruvate out of this equation. But the pathway that pyruvate takes once it is produced from glycolysis does differ when we don't have any oxygen present in our environment. So we'll start with the probably the least well known of the two processes that we're going to discuss, and this is anaerobic respiration. Anaerobic respiration is an alternative pathway that can be used to make a lot of ATP, at least more so than glycolysis, but it's going to use another molecule to accept those final electrons instead of oxygen gas. Now, a lot of the time that is going to be sulfate. So this is the pathway that organisms use when they live in environments where oxygen gas is not present all the time, basically. So this is going to follow a similar electron transport chain like we see in aerobic cellular respiration. But at the very end, when we send off those electrons after they've progressed through the pathway, oxygen gas isn't going to be there to pick it up. Instead, it's going to be a molecule like sulfate, SO4. Now, the electron transport chain does show up in both aerobic and anaerobic respiration, but not in the alternative process of fermentation. So fermentation will be what we look at next. So fermentation is what will allow our cells to create a continuous production of ATP only using substrate level phosphorylation and glycolysis. And then we have to regenerate those electron high energy carriers. So we have two variations of fermentation by which we can regenerate these high energy electron carriers, and they're named so to describe what other product they're creating in this process. So instead of ending with pyruvate, we can either end in an alcohol in the form of ethanol, or we can end with a lactic acid molecule, otherwise termed lactate. So the first one of these that we're going to look at is alcoholic fermentation. Alcoholic fermentation is where we take the pyruvate and we convert it to ethanol via two steps. So we first release carbon dioxide from the pyruvate, and this forms an acetaldehyde. So we get carbon dioxide out of this mix, and then the acetaldehyde is reduced by the NADH. It accepts an electron, and we form ethanol. So we get two products out of this pathway in addition to recycling our high energy electron carrier. We get an alcohol, ethanol, and we get carbon dioxide gas. It's for this reason that alcoholic fermentation is used to create breads and beer, other alcoholic beverages. We're producing that carbon dioxide that gives bread as it's rising, those bubbles. And ethanol, of course, can be our product used for alcoholic beverages. To compare alcohol fermentation to the alternative, which is what takes place in our cells, we have lactic acid fermentation. So this is where we convert, we don't have two steps, just one step. We convert our pyruvate to lactate, which is an ionized form of lactic acid. No carbon dioxide is being produced in lactic acid fermentation. Instead, we just have this buildup of lactic acid. So if you've ever been engaging in a lot of strenuous exercise, your muscles may start to ache. And that's because there is this buildup of lactic acid. We have an oxygen deficit, so we've had to make small amounts of energy without oxygen present via the process of lactic acid fermentation. We also use lactic acid fermentation to make some foods that we all enjoy, things like cheese, yogurt, and chocolate. Both use lactic acid fermentation to kind of cure the molecules and to make our food very delicious. I have one last animation from the textbook for us to round off this discussion of cellular respiration and the creation of cellular energy via a variety of processes. This one in specific is going to look at fermentation.
All cells are able to synthesize ATP via the process of glycolysis. In many cells, if oxygen is not present, pyruvic acid, is metabolized in a process called fermentation. By oxidizing the NADH produced in glycolysis, fermentation regenerates NAD+, which can take part in glycolysis once again to produce more ATP. The net energy gain in fermentation is 2 ATP molecules per molecule of glucose. Fermentation complements glycolysis and makes it possible for ATP to be continually produced in the absence of oxygen. 2 common types of fermentation are described here. Alcohol fermentation, which occurs in yeast, results in the production of ethanol and carbon dioxide. Lactic acid fermentation, which occurs in muscle, results in the production of lactate, lactic acid. Let's see the relationship between glycolysis and alcohol fermentation. Glycolysis produces NADH, ATP, and pyruvic acid. If oxygen is not present, NADH cannot be oxidized in the electron transport chain. Without fermentation, the cell would run out of NAD+, bringing glycolysis to a halt. In alcohol fermentation, the pyruvic acid from glycolysis loses 1 carbon in the form of carbon dioxide, and the product is then reduced to ethanol by NADH. With the formation of ethanol, NADH is oxidized and becomes NAD+. With a continuous supply of NAD+, glycolysis can continue producing more ATP. During fermentation, the NADH produced by glycolysis is oxidized, ensuring a continuous supply of NAD+ for glycolysis. Alcohol fermentation occurs in yeast cells. Let's observe the relationship between glycolysis and lactic acid fermentation. Glycolysis produces NADH, ATP, and pyruvic acid. If oxygen is not present, NADH cannot be oxidized in the electron transport chain. Without fermentation, the cell would run out of NAD+, bringing glycolysis to a halt. In lactic acid fermentation, the pyruvic acid from glycolysis is reduced to lactic acid, by NADH. With the formation of lactic acid, NADH is oxidized and becomes NAD+. With a continuous supply of NAD+, glycolysis can continue producing more ATP. During fermentation, the NADH produced by glycolysis is oxidized, ensuring a continuous supply of NAD+ for glycolysis. Lactic acid fermentation occurs in muscle cells.
And so, ladies and gentlemen, that is the end of our discussion on cellular respiration and our cell's ability to create cellular energy using glucose and other organic molecules. This will be a wrap on our 3rd unit, so I look forward to seeing you guys again in Unit 4.